Genes provide the instructions for making specific proteins, but a gene does not build a protein directly. The bridge between DNA and protein synthesis is the nucleic acid RNA. Getting from DNA to protein requires two major stages: transcription and translation. Transcription is the synthesis of RNA using information in DNA. Translation is the synthesis of a polypeptide using the information in the mRNA. Transcription and translation occur in all organisms. Genes program synthesis via genetic messages in the form of messenger RNA. Put another way, cells are governed by a molecular chain of command with a directional flow of genetic information. Messenger RNA, the carrier of information from DNA to the cell's protein synthesizing machinery, is transcribed from the template strand of a gene. An enzyme called an RNA polymerase pries the two strands of DNA apart and joins together RNA nucleotides complementary to the DNA template strand, thus elongating the RNA polynucleotide. Like the DNA polymerase that function in RNA replication, RNA polymerase can assemble a polynucleotide only in its 5' to 3' direction. Unlike DNA polymerase, however, RNA polymerases are able to start a chain from scratch and don't need a primer. Specific sequences of nucleotides along the DNA mark where the transcription of a gene begins and ends. The DNA sequence where RNA polymerase attaches and initiates transcription is known as the promoter. The stretch of DNA that is transcribed into an RNA molecule is called a transcription unit. The three stages of transcription are initiation, elongation, and termination. The promoter of a gene includes within its transcription start point and typically extends several dozen or more nucleotide pairs upstream from the start point. RNA polymerase binds in a precise location and orientation on the promoter, therefore determining where transcription starts and which two strands of DNA helix is used as the template. As RNA polymerase moves along the DNA strand, it untwists the double helix, exposing about 10 to 20 DNA nucleotides at the time of pairing RNA molecules. The enzyme adds nucleotides to the 3' end of the growing RNA molecule as it continues along the double helix. In wake of this advancing wave of RNA synthesis, the new RNA molecule peels away from its DNA template and the DNA double helix reforms. The mechanism of termination differs between bacteria and eukaryotes.
So today we're going to be talking about translation. In translation, a cell reads the genetic message and builds polypeptides from it. So in order for a cell to translate, we need a tRNA, which stands for transfer ribose nucleic acid. So they have two sites which an amino acid could bind to here, and a nucleotide triplet can pair here with a complementary base pair of the mRNA. So how do we know that transfer RNA picks which amino acids for certain codons, which are 3 segments of the mRNA? So in order for that to happen, the cell needs an enzyme, which we're going to call tRNA synthase. So this one has an active site which only fits a specific combination of amino acid, which is going to be played by our little green ball today, and tRNA. So once these are fitted into a enzyme, this tRNA gets charged. Once it is charged, it is released from the enzyme and then delivers amino acids to polypeptide ribosomes. So our ribosome consists of two parts. We have a little subunit of the ribosome, and then we have a larger unit of the ribosome. So this ribosome facilitates tRNA anticodons, which are going to be 3 segments right here on the bottom tRNA, which correlates to the mRNA codons of this transcribed strand. And the ribosome is only functional if it's attached to an mRNA nucleoid or molecule. Sorry. So this ribosome has 3 binding parts. You can see we have E, P, and A. So P is called the peptidal site, which holds the tRNA. We're going to put this, our little buddy here. And he holds this tRNA holding the growing polypeptide. So we have this buddy, our polypeptide that's growing because our cell is undergoing translation. Then we have an A site, which is this buddy right here. It holds tRNA carrying the next amino acid in the chain. So this tRNA is going to code for this amino acid due to the codon that is happening in this one, which contains 3. So the tRNA has 3 anticodons which signal for this amino acid. So placeholder A will be able to hold this tRNA for the next codon that is given by the mRNA. And then we have the E site, which is the site where discharged tRNA leaves the ribosome. A discharged tRNA is basically when it's not attached to a polypeptide or an amino acid. So as the ribosome brings all of these together, it initiates initiation factor protein, which brings polypeptide to build. So in place A, we have tRNA that places the anticodon with the codon with the amino acid. And then this tRNA, while that's happening, this tRNA gets discharged because it already added a covalent bond to it. So this gets transferred over to E, which is going to be ready to exit. The polypeptide chain is set there. We have this transfer RNA go to the P site while it adds a chain to this polypeptide, which is helped by a protein. And then we get another tRNA who gets ready for another amino acid to be read of its codons. So, and then by the end of it, it is a growing polypeptide. And then this gets signaled to its end, which a stop codon in site A can read it. And then this is where molecules of water attach to the polypeptide and transfer RNA is gone, which signals the termination of growing a protein. So we are left with a protein by the end of it.